Look at your hand right now. Hold it flat, fingers together, palm facing up. Compare your ring finger to your index finger. For most of human history, nobody thought this was worth a second glance. Then, in 1875, a German anatomist named Karl Ecker noticed something strange while dissecting cadavers in Freiburg. The length of the ring finger relative to the index finger was not random. It followed a pattern, and the pattern seemed to differ between men and women. Ecker wrote it down almost as a curiosity, a footnote buried in a much longer anatomical survey. For more than a century, almost nobody followed up on it. The observation sat quietly in old journals, while anatomy moved on to bigger questions. Then, in the 1990s, a British biologist reopened the file, and what he found set off one of the strangest and most contested chapters in modern human biology, a chapter that is still being written today, with real scientists still disagreeing about what your fingers are actually telling you. Here is why this matters right now. In the last 5 years, large genetic databases containing the DNA of hundreds of thousands of people have finally been searched for answers. Some of what they found confirmed old suspicions. Some of it overturned decades of assumptions. And a huge amount of what had been published about this trait in the meantime, in newspapers and pop psychology books, on breathless internet lists, turned out to be exaggerated, poorly tested, or flatly wrong. So before you decide what your own hand means, it is worth knowing the real story, the one built on replicated evidence rather than viral claims, because the honest version is stranger and more interesting than the myth. The trait itself has a name now. Scientists call it the second to fourth digit ratio, usually shortened to 2D, 4D. You measure the length of the index finger, the second digit, from the crease at its base to the tip. You measure the length of the ring finger, the fourth digit, the same way. Then, you divide the index length by the ring length. If the two fingers are exactly equal, the ratio is 1, 0. If your index finger is longer than your ring finger, the ratio is above. 1, 0. If your ring finger is longer than your index finger, the ratio is below 1, 0. That third case, a ratio below 1, 0, is what most people mean when they say their ring finger is longer than their index finger. And it is the case this video is built around. The person who turned this into a serious research field was John Manning, a biologist at the University of Liverpool, later Swansea University. In 1998, he began publishing studies proposing that the digit ratio was set before birth, fixed permanently by the hormonal environment inside the womb, and that it stayed essentially unchanged for the rest of a person's life. His theory, now usually called the prenatal androgen hypothesis, argued that between roughly weeks 8 and 14 of fetal development, a critical window when the fingers, the genitals, and several brain structures are all forming at once, the relative levels of testosterone and estrogen reaching the fetus subtly influence how long each finger grows. Higher relative testosterone exposure during that window, Manning argued, nudges the ring finger to grow a little longer. Higher relative estrogen exposure nudges the index finger to grow closer in length to the ring finger or past it. Because this happens so early and because bone proportions, once set, do not change with age or lifestyle, the ratio becomes a kind of frozen snapshot of a moment in fetal development that no memory, photograph, or medical record could otherwise capture. The most solid, most repeatedly confirmed finding to come out of this research is a simple sex difference. On average, across dozens of studies and tens of thousands of measured hands, men tend to have a slightly lower digit ratio than women. One of the largest cross-cultural tests of this came from a 2003 study led by psychologist Richard Lippa, who examined digit ratios in participants across 29 countries and more than 150 cities. The sex difference showed up almost everywhere he looked, small in magnitude but statistically consistent. This single finding, a modest but reliable difference between male and female averages, is the most defensible result in the entire field. And it is the finding that gave the prenatal hormone theory its initial credibility. The biological plausibility of the hormone theory got a boost from an unlikely source, a group of people with a rare medical condition called congenital adrenal hyperplasia, or CAH. In this condition, the adrenal glands of a developing fetus produce unusually high levels of androgens, the hormone family that includes testosterone, starting early in pregnancy. Researchers studying girls born with CAH who were exposed to elevated androgen levels in the womb despite being genetically female found that their digit ratios tended to run lower than average, closer to typical male patterns. This was treated as important supporting evidence, a natural experiment showing that prenatal androgen exposure really could shift the ratio, independent of a person's sex at birth. Animal research added another layer. In 2011, developmental biologists Martin Cohn and Zhengui Zheng, working at the University of Florida, ran an elegant experiment on mice. They found that the same genetic signaling pathway controlling the growth of the digits in the developing limb, a family of genes called the HOX genes, especially HOXA and HOXD clusters, also plays a role in shaping the genital tubercle. the embryonic structure that later becomes either a penis or a clitoris. By manipulating androgen and estrogen receptor signaling in mouse embryos, they were able to directly shift the ratio between the second and fourth digits in a dose-dependent way. This was a genuinely important result. It gave a plausible developmental mechanism, a shared genetic wiring between the hand and the reproductive system, for why hormone exposure and finger proportions might be linked at all. It is one of the reasons the hormone hypothesis is still taken seriously by parts of the scientific community rather than dismissed outright. But here is where the story gets complicated and where a lot of popular science writing quietly leaves out the messier parts. Once Manning's original papers came out, the digit ratio became one of the most popular variables in psychology and behavioral biology for the following 2 decades, and researchers began testing it against an enormous list of human traits: athletic ability, aggression, financial risk-taking, sexual orientation, fertility, musical talent, autism risk, sporting success in specific disciplines like football and running, personality traits, even facial attractiveness. A famous 2008 study by John Coates and Joe Herbert at Cambridge looked at digit ratios among London financial traders and reported that traders with lower ratios presumed higher prenatal testosterone, earned more money, and held their jobs longer during periods of market volatility. It became one of the most cited digit ratio papers in the popular press, endlessly repeated in articles about hormones and Wall Street. The problem is that as the field matured and sample sizes grew larger, many of these flashy correlations weakened dramatically or disappeared. Meta-analyses, studies that pool the results of many smaller studies together to check whether an effect is real, began delivering uncomfortable news. A large 2014 meta-analysis on digit ratio and athletic performance found the relationship, while statistically present in the pooled data, was far weaker than individual headline-grabbing studies had suggested, and highly inconsistent from one small study to the next. Research attempting to link digit ratio to sexual orientation produced a genuinely mixed and often contradictory picture, with some studies reporting small associations, particularly in women, and others failing to find anything at all once sample sizes were increased and measurement methods were standardized. Claims connecting the ratio to autism, to specific personality traits, to romantic success, and to general intelligence have mostly failed to hold up under rigorous replication. In 2020, a large-scale review published in the journal Early Human Development bluntly noted that a significant portion of digit ratio research suffered from small sample sizes, inconsistent measurement techniques, some studies using rulers and calipers on live hands, others using photocopies or scanned images, each producing slightly different numbers and a substantial risk of publication bias, where studies finding a striking result were far more likely to be published than studies finding nothing. Twin studies added another important thread to this puzzle. Researchers Martin Voracek and Marianne Dressler, working through the 2000s, compared digit ratios in identical twins, who share essentially all of their DNA, against fraternal twins, who share only about half. If digit ratio were purely a product of the shared prenatal hormonal environment inside one womb, identical and fraternal twins carried together should show fairly similar ratios to each other, since both types of twins share that same womb. Instead, the studies found that identical twins resembled each other in digit ratio far more closely than fraternal twins did, a pattern that pointed toward a substantial genetic contribution running independently of, or alongside, the shared hormonal environment. Some of these twin analyses estimated the heritability of digit ratio, the proportion of variation across a population explained by genetic differences, at somewhere in the range of 60 to 70%. A surprisingly high number for a trait originally framed almost entirely in terms of prenatal hormone exposure. This is part of why modern researchers now describe digit ratio as a trait shaped jointly by genetics and the hormonal environment, rather than treating those as competing explanations. The genes appear to set the stage, and the hormonal environment appears to influence how that genetic potential is expressed during the narrow developmental window when the fingers are forming. The trait is not unique to humans either, and that turns out to be one of the more persuasive pieces of evidence for a deep evolutionary origin. Researchers studying primates have found similar sex-linked digit ratio patterns in several other species, including chimpanzees and, in some studies, gorillas, with males in these species also tending to show comparatively longer ring fingers relative to index fingers than females do. Because these species diverged from the human lineage millions of years before recorded history, a shared pattern across species suggests the underlying developmental mechanism, the Hox gene and hormone receptor pathway identified in the mouse experiments, was already in place in a common ancestor long before modern humans existed at all. Some evolutionary biologists have proposed that the trait may be linked to sexual selection. The idea that certain physical features become more or less pronounced over generations because they are tied, even loosely, to mate choice or to hormone-linked traits that influenced survival and reproduction in ancestral environments. This remains a genuinely open and actively debated question. Not a settled conclusion, but it is a serious hypothesis discussed in the peer-reviewed literature rather than a piece of internet folklore. There is also a more basic measurement problem that rarely makes it into the popular coverage. Digit ratio is small. The typical difference between a male average and a female average across large studies is only a few hundredths of a single ratio point, something like the difference between an average ratio of 0, 98, and 1 of 1, 0, 0. That is an extremely small signal to extract reliably from a measurement taken with a ruler on a living hand, where soft tissue, finger positioning, and even the pressure of the measuring instrument can shift the reading. Researchers who use precision calipers or high-resolution photographs get more consistent numbers than researchers using simple hand measurements, and that inconsistency across studies has made replication genuinely difficult. Some biologists have gone further and suggested that even the underlying hormone theory itself needs re-examination. A widely discussed 2015 study led by researcher Gareth Richards attempted to directly measure prenatal hormone levels using amniotic fluid samples collected during pregnancy, then followed those same children after birth to measure their digit ratios. The direct correlation between measured prenatal testosterone and later digit ratio was weak and, in some analyses, not statistically significant at all, a result that surprised and unsettled parts of the field, since it had been the central untested assumption holding the whole theory together for almost 2 decades. So where does that leave things? The honest scientific position in the mid-2020s looks something like this. The sex difference in average digit ratio is real and has been replicated many times across many populations. So something biological is clearly happening. The animal research on Hox genes and hormone receptor signaling shows a plausible, elegant mechanism by which prenatal hormones could shape both the reproductive system and the fingers using shared developmental machinery. But the leap from that established mechanism to using an individual person's digit ratio as a meaningful predictor of their personality, their abilities, their sexual orientation, or their future success has not held up. The trait is real. The extreme claims built on top of it mostly are not. The digit ratio field also became a useful case study in how quickly speculative science can spread during a crisis. During the early years of the COVID-19 pandemic, several preliminary papers proposed that lower digit ratios, and by extension higher prenatal testosterone exposure, might correlate with more severe COVID-19 outcomes, based on the observation that men were being hospitalized and dying at higher rates than women in many countries. The hypothesis got picked up quickly by science writers looking for an explanation for the sex gap in outcomes. But the studies behind it were small, often based on limited hospital samples, and were quickly criticized by other researchers for weak methodology and for mistaking a correlation between two things, both linked to sex, digit ratio, and infection severity, for a direct causal chain between them. Larger, more careful follow-up research failed to establish a reliable link, and the hypothesis faded from serious discussion within a couple of years, replaced by explanations rooted in more established differences in immune response and pre-existing health conditions between men and women. It is a useful example of how a real, previously documented trait can get pulled into a new and unrelated headline the moment a plausible-sounding story is needed, regardless of whether the underlying data can actually support it. Partly in response to years of criticism over inconsistent measurement, later digit ratio researchers pushed to standardize how the trait is actually measured. Early studies varied wildly, some using a simple ruler pressed against the palm, others using photocopies of the hand, others using digital calipers accurate to a fraction of a millimeter, and still others using X-ray images of the bones themselves rather than the visible finger, which sidesteps the problem of soft tissue and skin folds distorting the measurement. Comparisons across these methods found that they do not always agree closely with one another, which explains a meaningful portion of the inconsistency that has plagued the field. X-ray-based measurements, which capture the actual bone length rather than the visible finger length, are now generally regarded as the most accurate method available, though they are far less practical for large population studies than a quick caliper measurement or a scanned photocopy. Researchers publishing digit ratio studies today are expected to report exactly which method they used, and to account for the fact that different techniques can produce ratios that are not directly comparable to each other, a basic methodological standard that much of the earlier, more sensational research simply did not follow. This brings us to the newer and more genuinely fascinating layer of the story, the genetics. As genome-wide association studies, often called GWS, became a standard tool in human genetics over the last 15 years, researchers finally had the technology to ask a more precise question. Rather than just measuring hands and guessing about hormones, they could scan the DNA of huge cohorts of people, sometimes drawing on biobanks with hundreds of thousands of participants, and search directly for genetic variants statistically associated with digit ratio. What these studies have found is that digit ratio, like height, like most complex human traits, is highly polygenic. It is not controlled by one gene or one hormonal switch. It is shaped by the combined small effects of many hundreds or even thousands of genetic variants scattered across the genome, each contributing a tiny nudge in one direction or another, layered on top of environmental and developmental factors in the womb. Some of the strongest statistical signals in these studies have landed near genes in the Hox family and near other genes, known to be involved in limb bud formation and skeletal growth during early embryonic development, which lines up neatly with the mouse research from over a decade earlier. This is one of the genuinely satisfying moments in the story. A case where independent lines of evidence, developmental biology in mice and population genetics in humans, arrived at overlapping conclusions from completely different directions. What the genetic research does not support, however, is the idea that digit ratio can be used to trace a person's ethnic ancestry or to identify which ancient population they descend from. It is true that when researchers pool digit ratio measurements from around the world, small average differences do show up between populations, the same way small average differences show up in almost every measurable human trait, from height to blood pressure to lactose tolerance. But these population-level averages involve enormous overlap between groups. Two people picked at random from any two populations on Earth are far more likely to be similar in digit ratio than different, and the specific genetic variants involved are shared across every human population studied so far, differing only in how common they are, not in whether they exist at all. No credible geneticist working in this field treats digit ratio as an ancestry marker, and consumer claims suggesting that a longer ring finger reveals a specific ethnic lineage are not supported by the peer-reviewed literature. This matters enough to say plainly because it is one of the most common distortions of real digit ratio science circulating online, taking a genuine, modest, population-level statistical pattern and turning it into a confident individual-level claim about heritage that the data simply cannot support. There is a broader and older story underneath all of this, one that goes back long before Manning, long before Ecker, long before any of this had a scientific name at all. Renaissance anatomists sketching the human hand noticed variation in finger proportions centuries earlier, treating it the way they treated any other observable difference in the human body, worth recording, not yet worth explaining. Classical Greek and Roman writers left scattered informal remarks about hand proportions in their descriptions of physical beauty and physiognomy, the now discredited practice of trying to read character from facial and bodily features. It is a useful reminder that humans have been staring at their own hands and looking for hidden meaning in them for thousands of years, long before DNA, long before hormones were even discovered as a concept. The digit ratio story is, in some ways, simply the most recent and most scientifically rigorous chapter of a very old human habit, the search for a physical signature that explains who we are. What makes the real version of this story worth telling is not that scientists found a hidden code written into your hand that reveals your fate, your talents, or your bloodline. It is that they found something more modest and, in its own way, more remarkable. A small, measurable physical trace of a developmental process that happens before you are even born, one shaped by ancient genes that predate humans entirely. Genes doing essentially the same job in the limb buds of mice, in the fins of fish millions of years ago, and in your own hand today. The Hox gene family, the genes now believed to play a central role in digit ratio, are some of the oldest and most deeply conserved genes across the entire animal kingdom, responsible for body plan patterning in creatures separated from us by hundreds of millions of years of evolution. When you look at your ring finger next to your index finger, you are looking at a visible outcome of a genetic and hormonal process, with roots stretching back far deeper than any human ancestry, deeper than any single population, deeper than the species itself. That does not mean the trait is meaningless. It means the meaning is smaller, more honest, and arguably more interesting than the version sold in listicles and thumbnail bait videos. If you have a lower digit ratio, a ring finger that reaches clearly past your index finger, the most defensible thing science can currently tell you is this. Statistically, across large populations, that pattern is somewhat more common in men than in women, a difference likely connected in part to prenatal hormone exposure, and in part to a long list of genetic variants inherited from your parents, variants involved in the same ancient developmental pathways that build every vertebrate limb. It does not reliably predict your personality, your athletic ceiling, your career success, your sexual orientation, or your ethnic background, no matter how many confident articles claim otherwise. Researchers are still working out exactly how strong the hormone link really is, still refining measurement techniques to reduce the inconsistency that has plagued the field for years, and still mapping which of the many genetic variants involved matter most and how they interact with each other and with the prenatal environment. There is also an important lesson buried in how this research unfolded, one that applies far beyond hands and fingers. Digit ratio became, for a while, a kind of magnet for weak science, the perfect trait for generating headlines, easy to measure with nothing more than a ruler, easy to compare across huge groups of undergraduate psychology students who happened to be sitting in the room, and tempting to link to almost any behavior a curious researcher wanted to study. Some of that research was careful and has held up. A great deal of it was not, built on small samples, unadjusted for multiple comparisons, and published because a positive, surprising result is simply more publishable than a boring null one. The digit ratio story is as much a case study in how science actually works, messy, self-correcting, full of early excitement followed by years of harder scrutiny, as it is a story about fingers. So here is where the science genuinely stands today, stripped of the exaggeration. Your digit ratio is very likely influenced by hormone exposure during a narrow window of fetal development, though the exact strength of that link is still debated and has been challenged by direct measurement studies that failed to find as strong a connection as originally assumed. It is shaped by a large number of genetic variants working together, many of them tied to the same ancient developmental genes that pattern limbs across the animal kingdom, genes with a history stretching back hundreds of millions of years before humans existed. It shows a small, consistent average difference between men and women, replicated in dozens of countries. It shows small average differences between broader population groups too, differences of the same modest, overlapping kind found in countless other human traits, not a reliable marker of any individual's specific ancestry. And it does not, despite 2 decades of eager claims to the contrary, reliably predict who you are, what you're good at, or what your life holds. The next time you glance down at your hand, at that quiet, permanent proportion between 2 fingers that was fixed in place before you ever took a breath, you are looking at a genuinely real piece of biology, a visible trace of an invisible process, one still being carefully mapped out by geneticists, developmental biologists, and endocrinologists working with better tools and larger data sets than anyone had access to even a decade ago. It is not a fortune written on your skin. It is not a secret code revealing your bloodline or your destiny. It is something quieter and, once you understand it properly, more genuinely fascinating, a small physical echo of the same ancient genetic instructions that once shaped fins into limbs, limbs into hands, and hands into the very fingers you're looking at right now, still carrying forward a developmental story that began long before there were humans to measure it at all. That, and not the myth built on top of it, is the story worth remembering. The next time someone tells you your fingers can reveal more about you than the careful, patient, decades-long work of actual scientists ever claimed they could,